exactly.
So let's break this down a little bit for people and give them a sense for how you study this, and then help reinforce some of these ideas and make this a little bit more grokkable for people. So let's just start with some common experimental setups in your world, or the world of hippocampus and memory and navigation. So we talked about place cells in the hippocampus. What are these, and what is the type of experimental configuration that they were discovered in?
So interpreting history is very difficult. One of my closest friends is John O'Keefe's in science. And I remember his first interpretation, how place cells were discovered, but his later interpretation is different. And I believe in the first story that he told me, which is that he was looking for place cells because of Tolman's ideas that animals are not automatons. They are not Skinnerian robots. They are not Pavlovian robots. But in fact, they have goals, and therefore they can relate themselves to made up goals. And in order to do that, they have to have they called it a map, initially a spatial map, but then he said it's not necessarily spatial, let's call it a cognitive map. And so John O'Keefe was looking for the instantiation of this in the brain. And he found it in the hippocampus. Now, the reason why I'm venturing to say that, and then the historians will figure out the details, is because if you go back to the discovery paper, the 1971 paper, I would say nobody but O'Keefe would conclude from those data that those firing patterns actually place fields. Because, again, please forgive me, John, but if you look at the figure details, there is nothing really tell you, and it's very difficult to observe that you put back the same animal at the same time, even though he was listening to the neurons, and so when the animal comes back to the same place, it fires again the same frequency. It's difficult to convince yourself that the animal was not doing some specific movement, or it was not looking at something specific.
So what you're saying is, if you go back to the original place cell paper, so for those who don't know, you put a mouse or a rat in a maze, and there's different places spatially in the maze, and when it goes to the same spot, the same neuron fires in the same way. One way to interpret that is that it's firing because it's representing that place. But you're saying, potentially, if you look at the same data, you could say, well, maybe, every time the rodent goes to this arm of the thing, it has a tendency to, make a certain movement because it's being provoked by something in the environment. And it's not actually a representation of space. It's a representation of some action taken at that place, or something like that.
Exactly. So that mistake, quote unquote mistake, has been done repeatedly over and over again, that people have described place fields in any part of the brain, from locus coeruleus, from the VTA, the olfactory cortex, everywhere, because this sort of constellations of activities occur unless you do many controls. So the reason why that paper is a prime example of a reflection of an extraordinary mind, because despite the fact that almost nothing holds in that paper, everything holds in terms of the hypothesized function of those cells. And everything that predicted there was confirmed again and again and again. That was the vision. And the reason why the vision there, I think, was because this is such a rare thing in science, that you have a preconceived idea and you actually find it. Typically, we find somebody most of the time we find things by accident.
I love how you're talking about the history of thought behind all of these episodes in history. What you're saying is he really came into these experiments with a strong preconceived notion of how things must work, and he took that seriously despite alternative interpretations of the same data.
And it's so rare in science. It's so rare. And he made another fundamental discovery also. Again, it's my interpretation. He already had a preconceived idea. This was called the phase precession. He was looking for a pattern. he didn't have the computational abilities of today. He had a storage oscilloscope that he and a few of us had, and then he found one run and said, there it was. The place 1, place 2, place 3, and there is phase 1, phase 2, phase 3. Is there. And so he's an extraordinary mind because he can look into the brain and find something that fits the right picture. In most of the time, it's the opposite. We never find what we are looking for. Instead, we find something, and I forget who said that the most important thing in a science laboratory is to say, Richard Feynman probably, that it's funny. It doesn't fit. Let's investigate it because it seems to contradict everything I knew before. This is the typical entry to a big theory, rather than the other way around. Okay, so this was the kind of thing, and then it, of course, and again, a very unusual thing that it wouldn't happen easily today. That you have a half job, you don't have a lot of job security, and so on, and you decide to write a book based on this preliminary observation, and he had 2 or 3 more papers afterwards. But then the explanatory power was so good, and his preparation for this was already there. So he wrote this Bible, and then, of course, there was a small group of people who followed him. You can see, 2 decades passes, and there is only dozens of papers or so about spatial navigation. But then when things became a little bit better, especially with phase perception, I became interested in this whole problem, not because of space and time, which was too noisy for me, but because time and phase as a mechanism entered into the picture. Said, oh my God, it's an interesting thing. O'Keefe also discovered that it's not only the firing rate of the neurons, but the relationship between where the action potential occurs on the phase of the theta cycle in a 100 millisecond time window is telling the rest of the brain, or at least to the experimenter, where the animal is. So you take a 100 millisecond time snapshot of activity, and you can precisely tell where the animal is. And then it's an interesting thing that something inside the brain, inside the hippocampus, is organizing the activity at the time scale of tens of milliseconds that relate to the animal's actions at the several seconds time window. And I think this sort of findings, and that one after the other, is what made people attend to the hippocampus, for perhaps one reason that I can summarize with this metaphor easily. For computational neuroscientists, noise is the crutch of everything. Without noise, nothing works. So something externally has to be given to a network to do anything useful. For me, it was the other way around. The brain, especially the hippocampus deep in the brain, is a self-organized structure. It generates patterns, but that's not noise. Your noise is my signal. And what I could say, rightly, I would say, is that the history of the hippocampus is the history of denoising the brain.